All right, students, good evening. Today we're going to talk about AC23, the RT-PCR lab. It can be a little bit confusing, so we're hopefully going to walk through exactly what it is you're doing and what all of the reagents that you add to the tubes do. All right, so first of all, we are doing an RT-PCR, or reverse transcriptase polymerase chain reaction. We'll get into what PCR is a little bit later. Let's do reverse transcriptase first. So the first part of our experiment is in the introduction, and it says we will use mouse RNA to make single-stranded cDNA. Now, if you recall from our central dogma discussion, that normally in a human we take DNA, we transcribe that over to RNA, and then finally we translate that into protein. So that's our normal flow and our normal way to go. However, it might be beneficial to us in some ways if we could actually go back this way. If we could take some RNA that we already have and go back to turn it into DNA, we could then manipulate it or maybe even insert it into another organism. And so that's basically what we're going to do in this lab. We're going to use an enzyme called reverse transcriptase to take some RNA that we have and turn it back into DNA. Now, something we should get out of the way right away is if we have RNA, and now I'm talking about mRNA, and we've got a long strand of that, and we know that it ends in a whole bunch of A's because all mRNA has a poly A tail. So there's our mRNA with the poly A tail, and we know that this is made up by copying a DNA strand. However, we also know that there are originally, in the DNA strand, we have maybe a few introns, a few of the exons that we keep, a few more introns, and some exons, right? So maybe we have intron 1, intron 2, exon 1, exon 2, right? And so this might be the actual DNA strand of whatever organism we're talking about, right? And so we cut out the introns, we process those out, and we bring the exons down here. And so in our mRNA, what we actually end up with are only the exon. So we have exon 1 and we have exon 2. So if we were to take this mRNA that's here and already made in an organism and go back up to the DNA, we are only going to be replicating the exons into DNA. Right? We have no idea what the introns are from just this mRNA because the organism has already cut them out. Right? So we don't know what the introns are. So we're not going to make an exact copy of the original DNA. We're actually going to make an approximation. So minus the introns, we're going to make, we put a little C, we call it cDNA. So that's actually what we're going to make. It doesn't exist in the actual organism because in the organism it will have introns as well as exons. Right? So that's just kind of an aside of what we're going to do. So we're going to take mouse RNA, that's this messenger RNA here, right, from a mouse, and we're going to go and make a single-stranded cDNA molecule. Now again, this cDNA molecule, it does not have no introns. We don't know what the introns were, right? Those are already taken out. So we will then use this cDNA as a PCR template and amplify only the GAP-DH gene. All right, so GAP-DH is actually used in glucose metabolism. So it's a very important enzyme. So it's nice that we get to use it. All right, and we're going to talk about PCR a little bit later, but just for right now, this is really a DNA copy machine. It makes many, many, many copies of DNA. So we can do lots of experiments with the DNA that we have. Okay? All right. So let's go down to step 1. What we're going to do is we're going to take a 0.2 microliter microfuge tube and we're going to label it. And we're going to add some things to it. First thing that we're going to add is 2 microliters mouse liver RNA. Now this is total RNA. All right, so what we're going to get is we're going to get a tube, and it's got all different kinds of RNA in it. It might have messenger RNA. It might have, I'll use a different color, it might have a bunch of tRNAs floating around. Right, it might even have some ribosome subunits floating around. So maybe it'll have rRNA. Okay, so we're going to get a tube with a whole bunch of things. However, only one of these different kinds of RNA has a poly A tail, and that's mRNA. So the mRNA is the only one with a poly A tail. And we're going to use that to our advantage, right? So mRNA has a whole bunch of adenines off of the end. And what we're going to do is we're going to add some of this oligo D T23 primer. And this is going to bind to the poly A tail. Okay, so now we've got this whole tube full of all kinds of tRNA, but we don't want all of those tRNAs. We actually only want the mRNAs. All right, so we're going to add plus our T23 primer. And so what we're going to get in our tube now is we're going to get all of these mRNAs with the poly A tail. Okay, all of these. Now for simplicity, I'm not going to draw all of the other rRNAs and tRNAs that are in here, but they're still in here. They haven't gone anywhere. Just so my drawing is a little bit easier, I'm only going to draw the mRNAs. Okay, and so what we do is we add a whole bunch of these primers, these T23 primers, and they have a whole bunch of T's. And so all of these primers will bind to the A's. Okay, and so that's what we're going to do. In that step, we're also going to add a little bit of nuclease-free water. This nuclease will break down the mRNA, and that's not good, obviously. We don't want the mRNA to break down, so it needs to be special water that does not have any nucleases in it. And that's what we're going to use in our reaction to bring up the volume so everything can work. All right. So now we're going to denature the RNA mixture by incubating in the PCR machine at 70 degrees. Anytime you see denature, you should recall that from proteins, right? It means to break down or open up or something like that. And so what we're going to do is we're going to incubate this at 70 degrees for 5 minutes, because sometimes what happens is these individual mRNA strands, right, with the poly A tails, if it just so happens that they've got the right sequence, sometimes they'll bind to each other. And we don't want that. We want all of these mRNA sequences to be nice and open so they can accept our T23 primer. So this 70 degrees will make sure that all of those mRNAs are by themselves. All right, and then after that, we add a few more things to our tube. We add in our reaction mix, and in this reaction mix, we're going to have things like ATP to provide energy. We're going to have buffer, which is basically just special water. We're going to have some nucleotides so we can make new DNA, right? Things like that. That's all going to be in our new tube. We're also going to have 2 microliters reverse transcriptase. right? And so if you recall, when we go back up to the top, at the very beginning of the lab, we talked about reverse transcriptase polymerase chain reaction, right? So this is the key player in our lab. This reverse transcriptase is what is over here, and it allows us to go from RNA back to DNA. So remember, we've got a whole bunch of mRNA from the mouse in the tube, right? All the mRNA that the mouse has made. It is not just what we want right now, okay? So it is not selected for at this point. This is all of the mRNA that the mouse has in the lab. I'm sorry, in the tube. And so what we do is we're going to add this reverse transcriptase enzyme mix. So we're going to add some of the enzyme into our tube. And so we'll draw our tube down here. So here's our tube again. Same tube. Right? So we're going to have our poly A tails that are all in here. And of course, we added our primer T in green here. So the T's will bind to the A's. Right? And so then this reverse transcriptase in green, here, I'll circle it in green. Right? It will go back and it will run this way. And it will make a DNA copy from the RNA template. So now our tube has some very interesting things in it, right? The green part is DNA. And the black part on top is still RNA. So now we've got these things bound to each other, and one half is RNA and one half is DNA. And that's kind of cool, right? So we're going to do that for a while. And then what we're going to do is we're going to put that mixture in the PCR machine, right? And the PCR machine is going to run one cycle. It's going to run 42 degrees Celsius for 1 hour to allow the cDNA synthesis to occur. So this 1 hour in the PCR machine is going to be where this actually happens, where we make the DNA right there. Right? This strand is going to go along the RNA template and make a cDNA strand. And that will be day 1.